Okay, good morning. Today we are going to start our lecture on development biology. It's a development biology class for the CSIR NET, and we are going to discuss the different part, mainly the development or the famous concept in which various questions are coming in CSIR NET. So, it's the class of development biology and development biology in which we are going to initiate the lecture mainly about the various concept, mainly the mesoderm induction, how it's going to be induct mesoderm induction take place, and how the various scientists produce the different experiments about the to analyze that which particular component or the molecular mechanism is important for this particular mesoderm induction. So today we are going to initiate our lecture about first by the famous scientist by the Spearman, Hans Spearman. I want to introduce the various concept or the various experiment about Hans Spearman, who got the Nobel Prize. So we are going to start our lecture about the in the development biology. It's the Hans Spearman. Hans Spearman is the person who got the Nobel Prize in 1935. The Nobel Prize in 1935 about the he has introduced a regulative development, mean to say at the how the different derm layers, ectoderm, mesoderm and endoderm going to regulate with the different components, how it's going to regulate, how it's going to induce which part is going to produce which type of tissues, whether it's the neural induction or what type of induction it is there. So we're going to introduce the Hans Spearman. First thing, the topic of that particular study is the inductive interaction in regulative development. This would be the topic of our today's study or about the today class. In the CSIR, various questions are coming day by day in every June or December examination about this particular process and about what type of process it is and experimental procedures. Mainly the questions are coming from the experiments that how Spearman going to analyze that particular interaction or the regulative development, how they are going to introduce that particular part. First thing that I'm going to study is the amphibian axis for development. As we know, there is a two type of specification. For example, first is autonomy. Autonomous specification in which I'm not, or the tissue or the blastomere is not dependent on the around blastomeres, that what it going to be its fate. Second is the mosaic or the regulative or the conditional, in which there is the development, the fate of blastomere is chosen by the environment, by the cells that surround itself. So we are going to start our formation. As we know, we start about the amphibian axis formation. As we know, there is two type of amphibian axis formation, in which the axis, as we know about, if you analyze the that I have studied, I have told you in the Drosophila development, there are different axes: dorsal, ventral, left, right axis, posterior, anterior axis. These axes are totally dependent on each other. That I've said that amphibian axis formation is combines two type of development. First is the specification, mainly is the autonomous specification, and second is condition. These specifications are totally dependent upon the autonomous, also said as mosaic, development, and the another is a regulative. Regulative development. So with this process particularly, we are going to initiate that our study of Hans Spearman, that what is Hans Spearman study, how they are going to start their study about the different mainly about the interaction or the interaction in the regulative development. So we are going to study with the Spearman study that demonstrated that if we're going to first study about it is the Spearman demonstration of the nuclear equivalence and nude cleavage. And the nude cleavage, the study name is the experimental that we're going to study is the Spearman demonstration of nuclear equivalence. Spearman demonstration of nuclear equivalence. So what is this study is going to be? If in a 8 cell stage they have taken a newly fertilized 8 cell stage of a nude egg and study it's a 8 cell stage and find out that if we with the lasso, mean to say a particular it's a mainly a hair of a horse, if we bind this, if we mean to distribute this particular egg or a nude egg into 2 parts such that one is a egg 8 cell stage and 7 cell stage is distributed in between, such that and bind the structure with a lasso. So this consists of a nuclei and this is without nuclei. This another part is consist of nuclei, another part is without nuclei. If it's a 8 cell stage, but in the different arena, if there is a migration of one nucleus to the another in the 16 cell stage, first is the 88 cell stage, initiate. Actually, we take a nude egg and form a lasso in an 8 cell stage in which one part consists of a 7 cell with nucleus, another part consists of a 1 cell without nucleus. And if in the 16 cell stage, when they get divided, 16 cell stage, there is a division such that there is the cells get divided and another part, one nucleus get transferred from one side to another side. This nucleus get migrate. One nucleus get migrated from one side of a lasso to the another side. What would be the development? This is the experiment they want to introduce, want to study that if the particular cell has role in the development, a particular side is regulative or development, and the third stage after 14 days. It's not, it's a 8 cell stage, it's a 16 cell stage, and after 14 days, what would be the final thing that we get is there is a development, but in the second point, the development is not so much appropriate take place. So this is the simple experiment of an specimen in which first they're going to be divided the cell, the whole egg, into two parts, in which one egg part is consist of the certain cell, 7 cells, and one of a cell without nucleus. Then there is a after the 8th cell get divided into the 16th cell, the nucleus get migrate from another part to another part. Then after 14 days, when the development take place, the development is not normal in the half or in the half. So there is a cleavage take place, but after this experiment, what Spearman conclude? Spearman conclude that a single cell going to make the whole organism apart from the other cells. Like a single cell has the power to get divided or get developed into a whole organism without it. So by this experiment, they conclude that after the constriction take place by the lasso, when the nucleus get transported, after the nucleus transportation, there is the whole organism get transformed into the a single cell can able to transform into the whole organism. This conclude that the of the specimen that they conclude that the first cleavage or separating the mainly the dorsal side or the ventral side, nuclei continue to divide on the both side, but the future dorsal side of the embryo give rise to the normal larva, but other cells are not able to give the whole embryo. Mean to say that if the cell, for example, is divided into two parts, one is dorsal and one is ventral. Dorsal cell can able to give whole embryo without any constriction, but other side a normal embryo, a normal larva. But the other side, which consists of the ventral region, unable to give a normal larva but an unorganized tissue mass, which by Spearman give a name which is known as pouch stuck. Which is known as the belly piece. What the ventral side consists of, the divided part consists of mainly, the divided part consists of mainly epidermal cell, means the ectoderm, and mesoderm, mean to say blood and mesenchyme, and the third one is the ectoderm, sorry, endoderm, which is gut cells. Mean to say, when dorsal or ventral get divided, the dorsal cell has the normal going to form a normal larva, but the ventral side is not going to form the simple larva. It consists of the, according to Spearman, it is a belly piece which is known as Bostock. It consists of the epidermal cells, ectoderm, blood and mesenchyme, which show it is a mesoderm, and an endoderm piece which are gut cells. The missing part here, the missing part is the no dorsal structure. What are the dorsal structure? Mainly nervous system, notochord, and somites. So, if the cell divided into mean to say, in the two experiment there is a different formation. If the dorsal side or ventral side get extracted differently, that I have shown in the diagram, if the dorsal side ventral side get disoriented or get separated, there is a different model, or the organism can't able to get its particular, there is a missing of a structure. The part which get the dorsal structure, having the normal larva, having the dorsal structure, nervous tissue, somites, and the notochord. But the missing part in the ventral portion is the nervous system, notochord, and the somites. But the thing is that if we divided the whole 8-cell stage in which dorsal or ventral side gets separated, there is a different result. But if there is no separation of dorsal and ventral side, and both the particular system or both the part get equally dorsal side, the organism is same as the one part or the another part. This particular show, what the experiment show, actually development biology is totally dependent on the experiment, and this experiment is going to help us to analyze the various part. Now we know that dorsal or the ventral side, when get separated, there is a different formation of stages, or the but organism development is very much different in the both sides. Mean to say, what we get that dorsal side is an important part for the structure determination or for the formation of certain dorsal structure, which is known as notochord, somites, and the nervous system. So we are going to initiate, mean to say that the dorsal side is important, the first inference that we get, and if that is absent, there is absence of a structure. So now the rules tells us that why there is a different result in the two different experiments. Same experiment, but the sites are different. So what would be the mainly inference we get? So the inference of that particular structure we get is that maybe when they get divided, the cytoplasmic substance, which are the determinants of that particular, the cytoplasmic determinants which are important for the development of particular cell, then we can say that I'm writing the inferences that we get from two different experiments. First, when the egg was divided perpendicular, on first cleavage plan. Cytoplasmic side is not equally distributed that we get in that particular organism. Mean to say, we can say that cytoplasmic was not equally distributed. So this is the we can say we are not I'm not telling you about the dorsal or ventral side. That maybe when the division or the lasso get bind, the cytoplasmic determinant get divided unequally at both sides, and due to this unequally distribution of the cytoplasmic determinants, there is a different different developmental, or the formation of certain structure get missing in one part as compared to the another part. This is the one inference that we got. But as we know that there is a certain part in the dorsal side, we know about the entry of the sperm going to form that when the first cleavage takes place, the part that is important is the gray crescent. Gray crescent is going to divide it equally into two blastomeres, and after its division, they're going to be equally divided into the two blastomere stage. Then in the fourth, and then the eighth, the process going on. But now, the specimen analyzed by another experiment, according to which if the part that get gray crescent as compared to other part are able to divide or are able to get all the structure as compared to the other side. So they could tell that if the particular cell, specimen example, there is a blastomere get divided and one part get the gray crescent area, it is the gray crescent. This gray crescent would mainly dominate that this part is going to be a normal larva, and this would not normal. This tells that the main dominant or the main regulative development or the main regulative role played by the gray crescent area, because it formed the normal area. Now the mainly it's a story. With one experiment, we're going to analyze, or we're going to find out a broad spectrum, and we want to analyze we have to narrow down each and every experiment inference or the result. Now the gray crescent area, we find out that the gray crescent area is important, and the part which has the gray crescent area is going to be normal, and the part which is not going which do not have the gray crescent area is not normal. I'm going to explain you by a simple experiment that I have said you, or through a diagram, but by a single experiment I'm going to give you a brief idea of the gray crescent. For example, this is a division, and this is the another one. Now when there is a first cleavage, they get divided into two parts, in which divided into a two part. Both are normal. This is A, this is B. This is the normal cleavage, first cleavage that take place. After the first cleavage, the gray crescent get divided into a both part equally, but here the gray crescent, here gray crescent, another one. In the B part, you can see the gray crescent is concise only to the one part, but in the A part, the gray crescent is divided equally into the two part. This show what would be the final result we got. In the two blastomere, if we divide these two blastomere into two part, there is a normal development. We get a normal embryo development. But it's a normal embryo development, so there is a normal development take place in the both part. But in this, what would be the final result? There is a belly piece or a normal development take place. So there is a belly piece and there is a normal development. This shows that there is a formation of belly piece and there is a normal development which have the gray crescent. Mean to say, the broadly by through the experiment we can tell you that the gray crescent is an important part, and the part which have the gray crescent, which the fate of that part is the normal development, and the another part which is going to form a belly piece. Now what is the gray crescent and what does it consist of? The second question. The story continues. In the story, if you analyze it through a story, you can able to grab it easily. But if you not, so you can get confused with the various examples, because development is the consists of the various experiments and examples and the different morphogenic factors that are important. We now know that the cytoplasmic determinant, the first thing, if we bind through dorsal ventral side, the structure, the experiment result is different. Bind to the anterior posterior side, the structure is different. If we separate the gray crescent area, the structure is different. Because up to that we know that the gray crescent is an important part, and the gray crescent would determine the particular development of that particular blastomeres. Now we know that gray crescent is going to determine or rise to the dorsal lip of the blastome, which is important for the gastrulation and formation of head, endomesodome, and notochord. So the interaction cells that important for the gastrulation is done by the dorsal lip of blastomere, which is formed by the gray crescent area. The precursor cell is the gray crescent. It's going to form the dorsal lip. Dorsal lip is going to important for the gastrulation, and also it is important for the head, endomesodome, and the notochord area is formed by the gray crescent. Now we know about the gray crescent, that there is an importance of gray crescent, and the gray crescent would determine the different part, mainly the dorsal part or the dorsal lip formation. So now we easily determine that what type of structure is going to be demonstrated by the dorsal lip. Okay. Another statement that is important up to that particular story is that I'm going to tell you. Listen to this story and then determine the different stages. you can able to easily crack CSIR net question, because CSIR net questions are coming from these stories itself, these experiments, and the full-on story in section C part is this story part, this experiment part will determine the particular questions. Next thing that is important is by another experiment they also find out that cells of early gastrula were uncommitted but of late gastrula is determined. How can Amphibians said that particular statement? What do you mean by uncommitted and what do you mean by determined? Early gastrula stage, the cells are uncommitted. Mean to say, when we shift these cells to another part, they are uncommitted. They can act as the cells with this surrounded. I'm going to tell you by experiment also, but you should understand first. In the late gastrula stage, when they get transferred from one part to another part, they are determined. They have to form the particular type of structure or the particular cells that they are determined. But mean to say, they are not controlled by the surrounding cells. They get divided according to its own will, according to its own structure. What would be the example of that particular part? First, I am going to tell you with the experiment, then I'm going to show you a table. In an experiment, so we have to take an experiment on both stage, early gastrula as well as the late gastrula. Early gastrula. Now the early gastrula. Experiment. A. Transplantation in early gastrula. Early gastrula. Transplantation early gastrula. What type of transplantation it is there? In the early gastrula, if in the early gastrula stage the blastopore lip, I think so in my previous classes I have told you about the blastopore lip, endoderm, mesoderm, ectoderm that are going to form. Now the part is now we going to transform this part to another stage, similar stage in the early there is a transfer of that part. This going to form the presumptive neural ectoderm, presumptive neural ectoderm, and this going to form presumptive epidermis. Okay, what would be the final formation? So the green part in the first particular embryo in the early gastrula stage, not in the embryo, is the early gastrula stage. Gastrulation is the division of or the formation of ectoderm, mesoderm, endoderm, so that its fate can be determined. In the early stage, the green part we show is the presumptive neural ectoderm. It's going to form certain neural tissue. If I'm going to transfer this particular part to the second particular gastrulation stage, to the presumptive epidermis, what would be the final fate we get? Final fate we get that this is going to transfer into a particular part which are going to form a epidermis. What we going to form? We are going to form stage so here we get the cell going to form epidermis. Neural base. So in this stage, in this gastrula stage, when the transplantation done by a neural ectoderm part to the epidermis, the final destination of that particular cell is epidermis, not the neural part, because the experiment is done on the early gastrula, not on the final stage or in the late gastrula. So this, what type of specification is this? This is the what? This is the condition. Mean to say, it is totally dependent on the cells surrounding by these cells, as it's going to form the neural ectoderm, but then it transplanted to the another part which get surrounded by the cells which are going to form the epidermis. The new one, the new formation or the final part is going to form a epidermis. Now, if the transplantation done by the bee in late gastrula, what would be the final? Transplantation in late gastrula. Transplantation in late gastrula. Now, what would be the fate if they're late? So, I'm going to form a late gastrula stage, which is different from the early gastrula. So, this is the late gastrula stage in which certain part is going to form presumptive neural ectoderm. Neural ectoderm. Now we transplant this part to another late gastrula stage, this is the what? This is the presumptive